Welcome back to the deep dive. We take stacks of research and, well, we try to boil it down to what's really surprising and useful.
Yeah, the core insights.
Exactly. And today we're diving into something often hidden in plain sight. The uh the really pivotal role of diversity within a single species.
That's right. I mean, for years when we talk biodiversity, we think, okay, how many species? Count them up.
Right.
But what about the variation inside one species? The genetic differences, the functional ones.
Mm-hmm.
Our source today really tackles that head on. It's a fantastic 2019 meta-analysis, The Community and Ecosystem Consequences of Intraspecific Diversity. Alan Raffard was the lead author.
Okay, so our mission here is pretty straightforward. Let's unpack the study, the synthesis, and show you, our listener, just how deep the impact of this within-species diversity goes. It really affects the whole ecosystem's health and function.
It really does.
It's often overlooked, but the numbers here, well, they tell a pretty surprising story.
Absolutely. The traditional approach in ecology, the biodiversity ecosystem functioning framework, or BEF, it mostly looks at interspecific diversity, how many different species you've got.
A species count, yeah.
But this paper really highlights that intraspecific diversity you got. The variation inside, say, the oak trees or the grasses or even the fish. It's not trivial at all.
Right.
They point to research showing it can account for, up to a quarter of the total biological variability in some communities.
Wow, 25%. So if you're only counting species, you're missing a huge chunk of the picture.
You really are.
Okay, let's get into how they broke this down. Because intraspecific diversity sounds kind of broad. The researchers looked at two main facets, right? Two different ways to measure it.
Exactly. We need to define these clearly before we hit the results.
Okay.
So the first one they called intraspecific richness. Think of it like species richness, but scaled down to the population level.
So just the number.
Just the number. How many unique genotypes or phenotypes are actually present in that group of organisms? 10 different genetic flavors of grass, or are they all basically clones, that kind of thing?
Got it. Simple count. What's the second facet?
A second one is intraspecific variation. This goes a bit deeper. It's not just how many types there are, but the specific identity differences between them.
Ah, okay. So the qualitative aspect.
Exactly. Are these types actually functionally different? Does one grow faster? Is one better at cycling nutrients? Maybe more drought resistant? It's about what makes them distinct, functionally speaking.
Right, the quality, not just the quantity. Okay, that makes sense. So richness and variation, how did they actually test the impact of these?
Well, Raffard and the team did this massive quantitative synthesis. It wasn't just reading papers. It was a proper meta-analysis.
Crunching the numbers.
Crunching the numbers from 90 published studies spanning quite a period, 2000 to 2018. And they covered a really diverse set of organisms, over 50 species. Trees, plants, fish, arthropods, you name it.
So a broad base.
A very broad base. And crucially, they really leaned on experimental studies. They wanted to find genuine causal links, not just, correlations.
That's key. Okay, let's start with the findings for the first part. Intraspecific richness, the number of types. Ecologists kind of had a theory about this, didn't they?
They did, yeah. There was an expectation about the shape of the relationship between adding diversity and getting more function. And this paper basically confirmed it empirically across many, many different systems.
And what did that confirmation look like?
They found a significant positive relationship, but, and this is important, it was nonlinear.
Okay, not just a straight line upward.
No, definitely not. It followed a very distinct saturating curve shape. Statistically, the best fit was an exponential asymptotic model, which is just a fancy way of saying the effect climbs really fast at first.
Right.
And then it levels off, like pouring water into a bucket that's already getting full. You get less and less additional effect.
So you see that initial big benefit. That's the complementarity, right? Where different types do slightly different things, making the whole group work better together.
Exactly. They use resources slightly differently or resist different stresses.
But then, like you said, it plateaus. And what's really interesting is where it plateaus. How quickly does that happen?
That's the surprising part. Functional redundancy, where adding another type doesn't really add a new function, kicks in really fast. The paper showed the plateau is generally reached around just 4 to 6 unique genotypes per population.
Only 4 to 6. That seems incredibly low.
It is surprisingly low. Once you hit that threshold, adding genotypes 7 or 8 doesn't seem to give you much more bang for your buck in terms of ecosystem function.
Wow. That low number, 4 to 6, that's a huge insight, isn't it? Especially for conservation. It suggests maybe focusing on protecting those first few unique types gives a massive return compared to, say, worrying about the difference between having 50 types versus 100.
Precisely. The biggest gains are at the low end of richness.
But the thing that really stopped me in my tracks was the sheer magnitude of this effect compared to what we traditionally look at.
Ah, you mean the comparison with species richness.
Yes.
That was probably the headline finding for richness.
Yeah.
The paper found that the ecological effects they measured for this intraspecific richness were, statistically speaking, equal in magnitude to effects previously reported for interspecific richness.
Equal.
Equal. The estimated mean effect size, the literal value they used, was almost identical for both, around 0.13.
Okay, let's pause there because this is really important for you, the listener, to grasp. Let's translate that 0.13 equivalence. What does it actually mean?
It means that ecologically speaking, the functional difference between an ecosystem that has, say, 10 species versus one with only 5 species
Okay.
is statistically comparable to the difference within a single key species population between having them all be genetically identical clones versus having just 4 or 5 unique genetic types present.
So losing genetic diversity within one species can have an impact on the same scale as losing several entire species from the ecosystem.
That's what the data suggests. The impact of loss at the genetic level within a species is operating on a similar ecological scale. Well, it's a monumental finding, really.
Yeah.
It elevates the importance of intraspecific diversity dramatically.
Necessarily monumental. Okay, so that covers richness. Let's move on to the second, maybe more complex facet, intraspecific variation, the identity differences.
Right. This analysis was actually even more robust because they had data from 51 different species to work with, which helped refine some earlier, more limited estimates.
And here, there was some kind of baseline expectation, wasn't there? A null hypothesis about how big this effect should be.
There was, based on previous work, the sort of null expectation for the effect size of this specific variation was around 0.259.
Okay, about 0.26. And what did they actually find?
Well, the observed reality kind of blew that expectation out of the water. The observed effect size of intraspecific variation, the specific identity differences on community and ecosystem dynamics was huge. The ESOBs, they called it, mean effect size observed, was 0.521.
0.521, compared to an expectation of 0.259.
Exactly. Almost precisely twice as large.
Wow.
It shows that the specific functional traits and the differences that define these genotypes or phenotypes, they are incredibly powerful drivers of ecological processes. The identity matters profoundly, not just the count.
Okay, so if the effect of identity is that strong, double the expectation, were there any situations where it wasn't quite so dramatic? Did they find factors that sort of moderated this effect?
That's a great question. And yes, they did. They identified some significant moderating factors, things that influenced how strong the effect was.
Like what?
Well, the first big one was the trophic level of the species where the variation was manipulated.
Trophic level, so where it sits in the food chain.
Precisely. The effects were significantly stronger when the intraspecific variation was manipulated in primary producers.
Plants, algae, the base of the food web.
Exactly. Things at the bottom. It makes intuitive sense, right?
Right.
Variation in the energy source, the foundation, that's going to ripple up and affect everything else, potentially dozens of consumer species.
Yeah, that follows. Variation there has widespread consequences. What else moderated the effect?
The second key factor was the level of organization at which the effect was measured.
Level of organization, what does that mean? Ecosystem versus...
Yeah, exactly. The effects of intraspecific variation were significantly stronger when they were measured at the ecosystem level.
So looking at big collective processes.
Right. Things like the overall rate of decomposition, nutrient cycling, total primary productivity, those big bulk processes. The effects were stronger there compared to measurements made just at the community level.
Which would be things like changes in the abundance of certain species or local biomass shifts.
Exactly. Things about the structure of the community itself.
Okay, so that suggests the most critical impacts of this internal diversity aren't just about, shuffling which species are common or rare. It's about maintaining the fundamental engines of the ecosystem.
That seems to be the implication. Things like soil chemistry, nutrient turnover, these core functions are highly sensitive to within-species variation, especially in producers. And the paper did note the strongest specific effects related to nutrient cycling and how communities assemble.
Fascinating. So let's try and connect the dots here. What's the big picture takeaway from all this? It feels like it really shifts our perspective, doesn't it?
It absolutely does.
Hmm.
These findings reveal this pivotal large-scale role for within-species biodiversity and how ecosystems work. It gives us a direct, measurable link between genetics, something happening at a micro scale,
Right, DNA level.
and these crucial ecosystem services we all rely on, which operate at a macro scale.
Bridges that gap.
It really does. And the theoretical implications are pretty significant, too. The paper argues we need to expand our existing frameworks. We've had this idea of the species genetic diversity correlation, the SGDC, for a while.
Yeah, okay.
They're suggesting we need to move beyond that simple correlation to think about a more complex tripartite relationship.
Tripartite relationship. Okay, that sounds a bit like an academic mouthful. Can we break that down simply?
Let's try. Instead of just thinking about species diversity and genetic diversity as maybe linked, but separate things,
Right.
they're saying picture it more like a three-lane highway.
Mm-hmm.
Lane 1 is intraspecific diversity. Lane 2 is community structure, who lives there. And lane 3 is ecosystem functioning, what the system does.
Okay, 3 lanes.
And these 3 lanes are tightly linked. They run parallel. They influence each other constantly. And importantly, because most studies they analyzed were experiments.
Yeah.
It confirms that changing the intraspecific diversity in lane 1 can directly cause changes in community structure in lane 2, even if the external environment, like climate or soil type, stays exactly the same.
So it's not just that the environment forces both genetic diversity and community structure to change together. The genetic diversity itself can be the driver.
It can be a direct driver, yes.
That's a much more complex picture to model and predict, isn't it?
Yeah.
So given this complexity, what did the authors say needs to happen next? What are the big research challenges? Because just counting genotypes, like we discussed, clearly isn't enough anymore.
No, it won't cut it. The first challenge they highlighted is untangling those complex causal chains.
Okay, cause and effect.
Right. Is the intraspecific diversity directly affecting, say, nutrient cycling? Or does it first change which other species can live there, and then those community changes affect the nutrients? It's about direct versus indirect effects.
How do you even tease that apart?
Well, they suggest future work needs to combine powerful statistical tools like path analysis, with really cleverly designed experiments to map out those direct and indirect pathways.
Sounds complicated, but necessary. What was the second challenge?
The second one is about the scale of the experiments needed to really capture natural complexity.
How so?
Almost all the studies included in their meta-analysis manipulated diversity in just a single target species, like varying the genotypes of one type of grass.
Okay.
But in reality, ecosystems have multiple interacting species, all with their own internal diversity. So to really reflect nature, future experiments need to try and manipulate intraspecific diversity in multiple interacting species at the same time.
Whoa. So imagine manipulating the genetic diversity of a predator and its main prey and maybe a key plant they both rely on all in one experiment.
Exactly. That's the kind of complexity we need to start tackling experimentally. It's tough, but probably necessary to get the full picture.
Definitely ambitious. Okay, and was there a third challenge?
Yes, a final point about what we measure. Instead of just broadly measuring genotypic diversity or phenotypic diversity, they recommend focusing more precisely on functional traits.
Functional effect traits. So traits we actually know have an ecological impact.
Exactly. Things like leaf nutrient content or root excretion rates or tolerance to heat or salt-specific characteristics with known ecological consequences. Instead of just counting genotypes, let's measure and manipulate the diversity of these specific ecologically relevant traits.
That makes sense. It gets closer to the mechanism.
Precisely. It should give us a much more powerful and predictive understanding of how intraspecific diversity actually works its magic on ecosystem dynamics. They also mentioned needing more studies that directly compare the effect size of intraspecific diversity against other major drivers like climate change or human disturbance, which are often studied separately.
Got it. Okay, let's try and summarize the absolute key takeaway for you, the listener.
Go for it.
It seems to be this. Intraspecific diversity, all that variation hidden within a single species, isn't just some passive thing species use to adapt over time. It's an active, fundamental ecological force.
Force, yeah.
And it shapes the entire ecological world around us, operating with a magnitude and impact that's totally comparable to the effects of losing a whole species.
That really sums it up.
Yeah.
Which leads naturally to a final thought, perhaps something practical for conservation and management folks to mull over.
Okay, leave us with something to think about.
Well, given that the big benefits of intraspecific richness seem to plateau really quickly at just 4 to 6 genotypes, remember?
Right. That low number.
And yet, the overall effects of intraspecific diversity, including variation, are comparable in scale to interspecific changes. Where should we be putting our limited conservation energy and resources?
Hmm.
Maybe we need to think more about prioritizing the maintenance of at least a critical threshold of within-species variation, especially in key foundational species, rather than solely focusing on maximizing the total species count in an area.
So a shift in focus, protecting that internal genetic toolkit, not just the species label itself.
It could be a vital part of the strategy, especially when you factor in huge constraints like climate change and habitat loss. It becomes a crucial calculation: how do we get the most ecological resilience bang for our buck? Focusing on that internal diversity might be key.
It's really quite something to realize how much ecological heavy lifting is being done beneath the surface, dictated by the subtle variations within species we see every day. Really makes you look at a field of grass differently.
It certainly does.
Thank you for joining us for this deep dive.